Good morning, everyone. I take this opportunity to welcome you again at the ISEE conference this year, the first one after 2 years of totally virtual meetings. For those of you who didn't come at the opening yesterday, is this welcome particularly addressed to? So, today is the first scientific session of our conference this year, and I am delighted to introduce our speaker today, who is Professor Frank Kelly. Professor Frank Kelly holds the inaugural Humphrey Badcock Chair in Community Health and Policy in the School of Public Health at Imperial College London. He is Director of the Environmental Research Group, Director of the National Institute for Health and Care Research, Health Protection Research Unit, in Health Impact of Environmental Hazards, and Deputy Director of the Medical Research Council Center for Environment and Health. He has substantial research activity which spans all aspects of air pollution research, from toxicology to science policy. A new area of investigation is ambient microplastics, where work is focusing now in their identification, detection, and potential health effects. Professor Kelly has published many peer-reviewed papers and many other things like books, proceeding papers, and so on, about toxicology and the health effects of ozone, nitrogen dioxide, and particulate pollution. He provides policy support to the World Health Organization on air pollution issues and is a member of the Health Effects Institute Review Committee. His talk today is entitled Airborne Microplastics. Are they the new enemy? So I'm really glad to give the floor to Professor Frank Kelly. Thank you, Frank.
Thank you, Claire, for that very embarrassing introduction. And good morning, everyone. It's really nice to be here. My thanks go to Professor Katziani and the organizers of ISEE 2022 for taking the brave decision to have this meeting in Athens when we can all get together again and catch up with old friends. So as Clea said, my first thing I need to say is that I'm talking about a subject which is quite new to the team that I lead in London, Airborne Microplastics. And to begin with, I really must give a big shout to these two individuals, Dr. Stephanie Wright and Dr. Joseph Livermore. Stephanie leads the microplastics team in the Environmental Research Group, and Joseph undertook his PhD under my supervision along with Stephanie. And really most of what you'll hear this morning is obviously down to their hard work. So first of all, before I start, as it were, complaining and dissing microplastics, we should remember that microplastics, or plastics in general, is very, very useful. It's had many, many benefits for society since its production in the early 50s. As you can see from this picture, it reminds us that it's very important in the food industry and has prolonged the shelf life of many products, which we take for granted these days. But of course, the other side of the coin is it's got many, many problems as well. And it's when we see it like this. I've just returned from my annual holiday, and I did see some of this on the beaches that I frequented, as I'm sure you all have. And this is now a common feature of many areas and countries around the world. So plastic debris is a rapidly emerging environmental issue, whether it be those plastic bottles and debris that we see on the beaches that we frequent, or if we look closely, whether it's these very small, what they're called plastic nodules that the plastic breaks down to. Some of you with very keen eyes can see that there are some here, just shown by the different color. They're just a little bit bigger than grains of sand. So they can break down into what we typically call microplastics. And that's defined by anything that's below 5 millimeters in any dimension. There's microparticles, as you see there, but there's also fibers, plastic fibers, either coming from things like fishing nets on our seas and oceans, or actually coming from the clothes that we wear. Increasingly, it contains synthetic material, and that synthetic material, it sheds billions of nylon or other plastic fibers when we put it through the wash cycle. So it's appearing in wastewater, it's appearing through the sewage system, and it's again passing into other environments such as waterways and seas. And then, well, not finally, but just as another example, here we have actually we make microplastics. These are tiny microparticles of plastic which are added to certain cosmetic products as exfoliants. We used to use natural products for this, such as coconut shell, but now the convenient way is just to use petrochemicals to produce plastics and break them down into these tiny microplastics, which we add to another product which we consume. So, as you probably all know, the Environmental Research Group in London is focused for many, many decades now, actually, on traffic-related pollution. That's really where our expertise and interest has been. But it was back, I guess, around 2013-14 when our interests started to turn away from, I suppose, exhaust particles, which we thought were increasingly coming under control through various new procedures and technologies, to the non-exhaust particles coming from traffic. And it was to my great surprise at the time when I started investigating these non-exhaust particles more, that whenever I looked at tires, I found out that a modern tire is more than 50% plastic. So this is a total change in the way that the tire industry has moved forward. And as you know, I mean, the number of vehicles on a road and the distance traveled by those vehicles has increased tenfold, certainly in the last 70 years. This is UK data, but it's pretty clear that this is the case really across the world. So we're having not only not more tire debris, but we're having more tire debris of a different composition over the last decade or two. So it could be, you know, it's a new issue and it could be something that's relatively important. So we decided to start investigating it along with others. And before we go into that, just here's a brief history of microplastics for those of you who hadn't focused on this area before. You can see that sort of mass plastic production began back only in the 1950s. So it's a one-generational issue, really. Around 72, there started to be reports of these plastic drifts in the oceans where the plastic debris was being brought together by various wave movements. In 2004, the term microplastic was coined by scientists at the University of Plymouth in the UK. I told you we started getting interest, but many others as well, around 2013-14 in tire wear. From 2014 onwards, there started to be publications indicating that you could find plastic being eaten by ocean life, either in shellfish, etc. In 15, it was reported to be part of the salt that we typically have on our dinner tables. So it was being contaminated, the natural salt that was being collected just with plastic nodules. 2016 it was first reported as atmospheric fallout, and I'll go into those studies in more detail. Then it was reported in indoor and outdoor air by various groups. It was found to be present in drinking water right across the world. And then in 19 we had the first study really which was related to humans. It was found in human feces. And since then we've had a number of studies, especially over the last 12 months, which have reported in lung biopsy tissue, in where else? In I guess, in blood more recently. So these are all single studies which are starting to come out and obviously need to be verified. But there is a real worry out there that we as a species are now being contaminated by this new form of environmental toxin. So, microplastics present in the air. The first study, certainly that I'm aware of, was presented in 2016. This was a study that came from Paris, and it was a very simple approach, really. What they did was they just placed rainwater collection vessels on 2 different university buildings. They left them there for a couple of weeks with the rain precipitation which was collected in these vessels. They also collected deposition depositing particles. And when they looked at these under the microscope, they found that, in fact, many of them turned out to be plastic in nature, either microparticles or fibers. But you can see the scale there. These are very large particles and obviously would not be respirable. So that was the first study in Paris, 2016. This was soon followed by a study from China, from Dongguan, where again, using the same approach, they collected rainwater samples from across the city, and they were able to detect a whole range of different types of microplastic. These consisted of different foams, fragments, polypropylene fragments, and polyethylene films. Then in 2019, we had a couple of studies. Bergman et al. Found airborne microplastics in snow samples in the Arctic, and then Klein et al. In the Bavarian Alps identified other airborne microplastics. But again, note here, these were relatively large, certainly from a respiratory point of view. But there is no doubt that they are present in the air, and they're being carried from source through to deposition. This was very much highlighted in Nick's study, which was published again in 2019 by Allen and colleagues, where they're looking in the French Alps. What they found was that on average there was 365 particles or fibers deposited in every square meter per day. If they size adjusted this down to something more realistic, excuse me, there was about 40 particles or fibers per square meter per day. And they also calculated that these were traveling a long way, up to 95 kilometers from source to deposition. So when we started on this work, we set ourselves a number of questions to address. We wanted to know if we could detect inhalable microplastics. If we could, we wanted to know what concentrations they were present in the air. We wanted to know what the dominant types were because, surprisingly, there's a whole multitude of different types of plastic present. Every product nearly that we use probably has some plastic component in it, different plastics. And also we were interested in whether these, if they are inhalable, whether they varied by location or not. Very straightforward set of questions. All the size questions obviously based on our previous knowledge about inhalable particles from PM10 downwards. So we wanted to know whether we could detect these inhalable microplastics below 10 micrometers. And if we could, then could we come up with an analytical method which actually identified what type of plastic that was? And maybe, as you'll see subsequently, what would be the chemical contaminants which that microplastic particle or fiber was actually carrying, either because it had been inserted at manufacture stage or because maybe it had picked it up during its journey in the environment. So we started off a pretty simple approach. We did the rainwater collection approach, as you can see here. But then we moved on quite quickly to this sort of multi-vial cyclone sampler, where we placed small amounts of very clean distilled water. Each of these cyclone samplers collected for a 24-hour period. So we had 7 of them in the sampler. We had a week's sample. we would take an aliquot, say 70 microliters out of the 100 microliter sample, and we would place that on a surface which we could then put under the microscope. So this was a casting method, as you can see here. And the method that we chose to actually detect the plastic component of the particles which we're collecting was initially Raman Spectroscopy, which again was a totally new area for us to work in. So very quickly we find that actually the substrate that you used to do your cast on was very important, because again the Raman spectroscopy was very sensitive to different substrates, and the one in which we ended up using routinely was a simple silver platform, so something like silver foil. And this was then give us a very clean background which we could then build our microplastic analysis on. So we would collect samples from the environment, either specifically using that multi-vial cyclone sampler, or using just the samples that we're collecting from the London Air network. We would spike those with other standards which we had produced ourselves, virgin plastics, and then we would undertake Raman spectroscopy on those. And you can see here some examples of that. And what we found was, in due course, we were able to work through to get the sensitivity sufficient to be able to identify microplastics of different types down to a cutoff about 2 microns. So we started pulling together data on an inhalable fraction between 2 and 10 microns of different types of microplastic. And we were able to identify the different types of microplastic by adding another technique, FITR, which allowed us to differentiate between the different chemical components of the plastics. So here we just see some of those spectra. So here's some, these are some environmental samples here. You can see that we're just picking up very, very small microplastic particles in these particular samples. We had a whole range of different virgin microplastics, which we could use as reference material. We could then build up the spectra for each of these, and by that we were able to then identify what we were collecting in our environmental samples. And so we built up what's called a plastics spectral database. You can see different types. This is polypropylene, polyethylene, PVC, etc. And you can see that in this green bit in the middle that they all have characteristic spectra. So you're getting a very clear and clean differentiation between the different types of microplastic particles or fibers that you're seeing. So with that sort of the spectral database in place, we then started to do a sort of ambient sampling program in London, where we collected size fractionated samples and put them through this analytical process. And we were able to determine both the number and types of microplastic particles there. And if we just look here at these two, this is from 3 microns up to 10 microns, you can see that they're all mostly the same type of plastic, but these are actually they have been heavily added to with a pigment which is often used in plastic manufacturers, this cupric coloring pigment here. But the important thing, I guess, which is not actually on this slide, but it's on the next one, is that it was these very small particles which were the ones that dominated in number. And as the particles got bigger in size, then the numbers went down considerably. So here's just a series of samples taken across London of the different types of particles that we found. There's polystyrene particle there, polypropylene particle there, and different other ones. You can see here there's fibers and there's particles being collected. So this is the bottom take-home line of that first big analysis that we did in London. The vast majority of microplastics that we identified were actually fibers and not particles. You can see the numbers here collected across a different series of samples. and the fibers really dominate by number anyway, the deposition rate per day. So when we did the calculations, we found that it was about, on average, 700 fibrous microplastics are deposited in every square meter across the city per day. Now, 700 doesn't maybe sound that much to you, but if you actually take a city the size of London, then that actually does add up quite significantly. Those are the different types. Polyacrylonitrile were by far the most dominant, polyester next, polyamide third. And when you add those all up, then on average, it's about 2 billion particles, mostly fibers, which are settling over the city every day. So that was our early work on ambient microplastics in the City of London and the techniques that were needed to be able to identify those. When we saw that a lot of these microplastics were actually fiber in nature, then of course our attention started thinking about, well, where are they coming from? Clearly a lot of it is coming from seems to be modern clothing or things like fabrics, coverings, carpets, etc. And so we decided to then start thinking about the indoor exposure to microparticles. And so we went into somewhere where there's quite a lot of friction going on, an indoor gym where people are, have a lot of synthetic clothing as part of their activities there. And we took our sampler into a gym environment, and we had set it up there. And again, what we have here is size fractionated microplastics here along the bottom, going from 1 here up to 400 micrometers. And you can see that, in fact, if we go up to the top here, in the respirable red box here, then we've got very high numbers of fibers compared to what we see as we move up through the size fractions. So there's a lot of fibers being released in a gym, an active situation in an indoor gym, and a lot of those fibers are very, very small. In fact, they're inhalable, and they're in the inhalable size range. So, to come clean with you now, as I've been telling you about methodology, which involved collection of your material, casting it on to a surface, putting it under a Raman microscope, and then using FTIR to analyze the type of plastic that's there. In reality, that is a very long procedure. And if you're actually going to just take one sample and analyze it properly, it could be up to 24 hours screening of that sample as the microscope moves across each part of it. And it also produces very, very large databases. So we realized quite early on that this was not going to be functional for moving forward with this technology. And so therefore, in the last probably 18 months, we've moved our analytical procedures forward to using pyrolysis, gas chromatography, mass spectrometry. And this is a much faster way in which to produce similar types of data. The equipment, unfortunately, is very expensive. We didn't have it to begin with. Fortunately, now I do have it. And the samples actually require very little workup indeed, because actually what you're doing with the pyrolysis step is just you're blowing everything apart. You're using 2D gas chromatography to separate all the components, and then you're using mass spectrometry to actually analyze each of the components. And it's actually the technology has been used, in other settings over the last decade, and it's quite mature now. Clearly, there has to be some work to do to get it working well for microplastics, but we and others are advancing that quite well now. So it's very targeted, it's very quantitative. You can do multi-samples over a 24-hour period. So I think this is the way forward if you're interested in this type of work. We're currently standardizing the workflows for analyzing all the inhalable particles from PM10 down to PM0.1, ultrafine or nanoplastics as they're now being called. And we've developed rigorous QA/QC validation methods as well. So really, going back to why are we doing this, and why have we spent the last sort of 5 years, as it were, pulling together the methodology to be able to address this question? Well, the big issue is we know they're there, we know they're not going away, but we need to know whether they're actually having an impact on health. This is an issue which is on everybody's mind working in this area. We also know that other particles, clearly those coming from traffic, have got, they have a well understood toxicological pathway involving oxidative stress, involving inflammation in the lung, etc., etc. And the big question then is just another type of particle which has got a different chemistry, is it got any similarities in that respect? The other thing, though, we need to remember is that if we're looking for the impact of these microplastics in our body, if it's not directly in the lung itself, but if it's beyond the lung, then we've got to appreciate that, in fact, we actually are we're ingesting a lot of microplastics, clearly every day, in the fluids that we drink and in the food that we eat. Every product that's been tested basically has got a microplastic aspect to it. Now, some of those studies are questionable, and they're questionable because it's very easy to actually contaminate a particle. In fact, we now have a clean room where we do all the work, and people are not wearing synthetic lab coats or anything like that anymore. So you have to be extremely careful when you're doing this type of work, especially if you're trying to quantitate, the presence in a biological sample or any other sample. But I think this is a major issue going forward, and one we're going to have to be very careful with if we're going to certainly get the inhalable side of things worked out properly. So what questions are we beginning to ask now we have our new techniques and technologies developed? So is microplastic toxicity influenced by the type or age of the plastic? Now, aging is very important. Because you don't get microplastics unless the plastic has been aged in the first place. That's how you get it down to that very small size. So actually, does that process of aging, which involves, sort of UV light, involves various chemical, sometimes breakdown or biological attack, or frictional attack then, does that actually change the toxicity of the particle? It may actually release some chemical components which were embedded deeply in the plastic before, for example. We want to know what are the most toxic combinations of plastics and microplastics. Clearly, it would be really nice to know if we could measure them in human samples. For example, we're looking in nasal lavage and induced sputum, etc., because we're interested in the lung. But there have been recent publications mentioned at the beginning of the talk where there have been indicated that they're present in human blood samples and fecal samples and in other lung tissue, for example, biopsies. But again, I would say the jury's still out in a lot of these studies because we need to make sure that the technology is appropriate, it's being used, and we're not getting contamination of those samples, which is a very simple step. And then finally, how does the exposure to microplastic particles influence their presence? i.e., do they bioaccumulate? And I think this is probably the real issue, because even though our exposure, even through inhalation, is probably quite small compared to other combustion-related particles, the question is, do they bioaccumulate? Can we remove them easily from our system? If we can't, then you can see that over time they could build up and they could then start obviously causing a dose-related response in a tissue sample. And then as we come near the end, a couple of slides clear, plastic is plastic, different types of plastic, but in fact to make plastic really useful in modern society, it's often the chemicals which are added to it, the pigments, the other chemicals which make them pliable or fire resistant. It's maybe those chemicals which are the real problem, and the plastic is just the magic bullet, the carrier into our bodies. So we need to appreciate that as well. Or heavy metals, which we already have a lot of worry about from a traffic point of view. So what do we have to rely on at this point in time? Well, there is some history out there. There is a condition called Flocker worker's lung. And this is a hazard which exists. It's an occupational exposure to this flock or dust, which some people are exposed to as part of the workplace. It leads over time to the development of a persistent dry cough, a shortness of breath, chest pain, all the classical sort of responses. On investigation, there's often infection in the airways and inflammation around the tissue surrounding the bronchi. And so this is a well-established occupational exposure to a natural fiber, as it were. And it's persistent and it's irritant in nature. So to finish, where are we? Well, we're at the beginning, I think, of quite a long journey. Just to remind you, plastic is now the third most abundant man-made material. There's been over 8 billion accumulative tons produced since 1950. It's not going to go away. It is a great product, but we've got to find out some better way of reusing it, or at least not letting it contaminate our environment. And the big worry I have is, in fact, plastic comes from the petrochemical industry. What are we doing to the petrochemical industry? We're trying to stop them producing petrol, diesel, gasoline, airplane fuel, etc. But they're going to need an output for their product. So the suspicion is that, in fact, we're going to see plastic production taking over a lot of that petrochemical use in the future. So if anything, it's going to increase. As I've already said to you, the microparticles fibers are present in the environment, these microplastics and now nanoplastics. It contaminates everything around us, the air, the water, the food. It's persistent, it's got hydrophobicity, it's got different shapes and chemical burdens. We know that there's now evidence of nanoplastic release, but this is really at the end of the investigated spectrum, actually getting down to those sizes of particles, but they do seem to be there. Much like, we're still struggling with the ultrafine particle issue. I think we're going to struggle with the nanoplastic issue for a long time to come. And it's unregulated. There's a global waste management crisis out there, which you're all very aware of. So what do we need? And this really goes, this is a message I'm putting out to maybe some of the younger people in the audience. If you're starting on your career and you want something interesting and hopefully useful to work on in the coming decades, then I think this issue of microplastics is a potential winner. We haven't got any data sets on the concentration of microplastics, the inhalable ones, other than, say, the few studies which I've talked about today. There's no published epidemiological studies linking to environmental microplastic exposure. So there's a challenge for all the epi people out there. And we lack toxicological data on different sizes, shapes, and types of plastic. So lots and lots of work to be done. Hopefully you enjoyed that, and I'm happy to take any questions you have. Thank you.
Thank you very much, Frank, about this very interesting introduction to an emerging problem that may actually occupy us in the future in terms of research. We have a little time for questions. Anyone from the floor who would like to ask a question, please? Yes.
Thank you. Very nice opening presentation and overview. I have two questions. What's your current best guess in terms of the future perspective? Is it the size of the particles or is it the surface characteristics? We have lots of studies on air pollution in general. We don't care too much about the surface characteristics. We still have relatively good results, I assume. Second question, are there any suggestions regarding markers of early biological effects, some inflammation markers that we could apply in epidemiological studies to move forward from the biomonitoring of exposure, like the nasal lavage, to adverse health effects? Thank you very much.
So it's a total guess, I'm afraid. I mean, size versus chemical composition, I think both are important. Ultimately, chemical composition may be more important, but obviously the size initially, it's to get to the target tissue. And then secondly, the issue about toxicology and the pathways involved, I guess. There have been animal studies done. As with historically with combustion-related particles, often the dose is too high. We don't know if it's realistic, but many of the classical pathways are activated, and you can show that different compositions will give you different levels of activation. And again, you know, oxidative stress, inflammation, these have all been recorded. So I don't think there's probably too many deviations from what we classically understand from particle toxicology, and it's really just the dose issue and the type issue, I guess, and the bioaccumulation issue. Yes.
Thank you for this amazing presentation, and I particularly appreciated your call to the younger generation for where we need to go in the next 20 years with this research. I heard a similar presentation about somebody who was following the ingested microplastic particles, and I was surprised that you didn't mention that many of those particles concentrate in the reproductive organs, both in women and in men. And I was wondering whether this was something that you haven't looked at, and then I just wondered what your comment would be to that. It was a very clear finding that the ingested particles were accumulating in the reproductive system.
Yeah, I mean, I've tried to be cautious with the literature. There is a study which mentions placental content of microplastics, and some of the images that are in that particular study, you know, we currently question. So I think, you know, when you have one study here, one study there, such as we got 2 studies in lung biopsies now, we've done our own work and we appreciate how difficult this is to do. So I just think the jury's still out. There's every possibility, of course, that they're right, but I think we need more evidence before we go overboard with those outcomes.
May I ask those who have questions to identify themselves, mainly for the virtual attendants? And there is a question from Tracy Woodruff. She says, doesn't the situation with microplastics remind one of other early environmental concerns, such as asbestos? Perhaps a precautionary approach is needed here? That's from the virtual participants.
Thank you, Clea. Well, you know, as many of us in the room probably think the precautionary approach should have been, you know, brought forward a long time ago for other exposures. It's just very, very difficult, I think, because if you just think about your own life, your own 24-hour normal day, and the plastic that you use and consume, you know, are you willing to give that up, or are you willing to pressurize whatever authorities to ensure that that plastic is disposed of or reused appropriately? It's just an enormous issue, which, you know, I just think realistically we're probably not going to deal with properly. I think, yeah, it would be great to use the precautionary principle at this point. But on the other hand, you know, I can't for one stand up and say we've got this solid evidence to say this is, you know, this is really detrimental to human health. I suspect it probably is, but I don't have that evidence. So maybe we need just a little bit more research in this area before we start shouting a little bit louder.
I think we can take one more question.
Okay. Hi, thank you, Frank. This is Joel Schwartz.
Where are you, Joel? Sorry.
Morning.
Okay.
So
yeah, all right.
I was wondering, when you showed us the inhalable fraction of the airborne plastics in London, it looked like, you know, the great bulk of them were coming from one particular type. And I wonder whether you were able to trace back what the source of that was. Because we want to know the sources if we want to know where to target our interventions.
Yeah, very good question. So we were doing our collections on the top of a university building at King's College on the Strand. So many of you know that's right in the centre of London. We did some windrows work to try and identify the sources of that. And basically, it was, as far as we could see, the main direction of source was coming from the city itself. So where there seemed to be, every day a lot of people moving. This is back in 2018-19 where the samples, I think, were collected. So there was a lot of high-density movement of individuals in the center of the city, and that seemed to be, where we were getting that major source from. So whether that then comes down to the clothing material of those individuals, or the buildings that they were moving in and out of. But that's the best I can say at this point. Thank you.
Thank you very much. I'm afraid we are running out of time. This was very interesting and very interesting questions, and I'm sure there are more. Frank, to thank you, we have a small appreciation.